these two hormones require a net flux, a net increase in the amount of intracellular calcium in order for them to work, in order for them to produce the response that they are expected to. As the last component of our discussion on secondary messengers, let's take a look into different hormones that uses protein kinases in their pathways. So there are two pathways that we are going to discuss. Those that are possessing inherent tyrosine kinase activity, and those that do not possess inherent tyrosine kinase activity. For those, or for the hormone possessing tyrosine kinase activity, we have insulin. Insulin, which is actually produced by pancreatic cells in response to hyperglycemia. Uh, so for the case of insulin, insulin induces or produces its physiologic response in coupling or in couple with activation of tyrosine kinase. On the other hand, the second pathway, known as JAK or STAT pathway, there's no integral or inherent tyrosine kinase, and instead, special associated proteins are involved. Let's take a look at the initiation of signal transduction by receptors that are associated to JAK kinases. For the case of enzymes like, or I mean hormones like growth hormone, prolactin, or cytokines and interferons, okay, they do not have tyrosine kinase. The receptors that are specific for them do not have tyrosine kinase. When the receptors are bound to their hormones, are bound to their effector molecules, the growth hormones, prolactin, or cytokine interferon, okay, the receptors dimerize, and an associated protein is actually phosphorylated. Okay, so as we are seeing here, the ligand bound to the receptor will result to the receptors to dimerize to so two receptors attaching to each other, dimer. Okay, and JAK-P, a active kinase, phosphorylates this receptor on its tyrosine residue. So it attaches phosphate on the tyrosine residues of the receptor. Okay, cytosolic proteins (STATs), okay, which are phosphorylated, will dimerize. Okay, and as they dimerize, because of the influence of the previous events, the or dimerized stat proteins, which are also phosphorylated, will start the process of translocating or transferring to the nucleus, binding to some specific REs, response elements, and the binding will result to the regulation of the process of transcription. So obviously, whatever is needed for the physiologic response to be produced as specific to the type of ligand, type of hormone that is recognized by the cell, will be the output, the output we expect from the process. Okay, so this ends our module number 3 in the mechanisms of hormone action. In the following module, module 4, we'll be examining thyroid hormones and growth hormones.